Good morning to all of you. Today we are going to discuss about one important biomolecule, amino acids. When we conduct the chemical analysis process, on any one living tissue or plant tissue or animal tissue, then we get two different type of fractions. The two different type of fractions are, one is a acid soluble fraction and the second one is a acid insoluble fraction. Acid soluble fraction has thousands of organic compounds, and acid insoluble fraction has thousands of biomacromolecules. In acid soluble fraction, biomicromolecules are present. In acid insoluble fraction, biomacromolecules are present. Acid soluble fraction also called filtrate. Acid insoluble fraction also called retentate. Amino acid is a biomicromolecule which is present in acid soluble fraction. It is an organic compound. All amino acids are alpha amino acids. Every amino acid has four valency positions. The four valency positions of alpha carbon occupied by four different groups. The four different groups of first carbon are hydrogen, free radical group, carboxyl group, and amino group. Carboxyl group, amino group, and free radical group are decides the physical and chemical properties of all amino acids. All amino acids are alpha amino acids because all essential groups are arranged at first carbon position or mth carbon. You know about the IUPAC name. In IUPAC name, the first carbon is alpha carbon, second carbon beta carbon, and third carbon omega carbon, etc. Among that carbon atoms. So amino group and carboxyl group are arranged at first carbon position or alpha carbon position. Due to presence of all groups in alpha carbon position, the amino acids are called alpha amino acids. Amino acids are building blocks of proteins or building blocks of polypeptide, because the other name of protein is called polypeptide. Amino acids are present in acid soluble fraction because their molecular weight is less than 800 Daltons. One simplest amino acid is glycine, and remaining two other simple amino acids are alanine and serine. Alanine and serine are two other important amino acids. All amino acids are substituted methanes because the important groups are substituted at mth carbon position. An organic compound having a amino group, carboxyl group on same carbon, then that all proteinaceous amino acids are called alpha amino acids. Based on the nature of R group, amino acids are divided into different types. The amino acids which are present in living organisms are two types. One are the non-proteinaceous amino acids and second one proteinaceous amino acids. The non-proteinaceous amino acids are approximately 170 and the proteinaceous amino acids are only 20. 20 amino acids are proteinaceous amino acid, remaining all other are non-proteinaceous amino acid. This is the number of non-proteinaceous amino acid and it is the number of proteinaceous amino acid. Three important examples given in your syllabus are, one is a glycine and second one is a alanine, Third one is a serine. This is a glycine, alanine, and a serine. In the structure of glycine, R group substituted with the hydrogen only. But in alanine with the CH3, in serine with the CH2OH. If we see the molecular formula of glycine as number of carbon atoms are only 2. So C2. Number of hydrogen atoms are total 5. So C2H5O2. It is the molecular formula of glycine. If we see the molecular formula of alanine is number of carbon atoms are 3. C3. And the number of hydrogen atoms are 3 plus 2, 5 plus 5 plus 2, 7. So C3H7O2. It is the molecular formula of alanine. And the molecular formula of serine is C3. C3 hydrogens are 3, 4, 5, 6, 7. And hydrogens are 7. O2, N. And here also one nitrogen atom is there. So C3H7O2N. And here also one nitrogen atom is there. Na? So because of this reason, C2H5O2N is the molecular formula of glycine. So C2H5O2N, C3H7O2N, and C3H7O2N. These all are the molecular formulas of glycine, alanine, serine. They are the simple amino acid with simple structure. In glycine, the R group is non-polar or hydrophobic. Non-polar or hydrophobic. But in serine, the R group is polar or hydrophilic. You know about the difference between polar and non-polar. Non-polar means present towards the inside, and polar means present towards the pole. Glycine is the simplest and smallest amino acid when compared with the remaining all other amino acids. All amino acids are levorotatory, but glycine is a dextrorotatory. Glycine forms porphyrin head in chlorophyll pigment. Chlorophyll pigment is a main pigment in photosynthesis process to trap the light. In that chlorophyll pigment, four pyrrole rings are present in the structure of a head. Each porphyrin head made up with glycine. Glycine also involves in the formation of heme protein. Heme protein is a structural component of hemoglobin, and also it is a part of cytochrome. Alanine involves in the formation of, alanine is another type of amino acid. It involves in the formation of coenzyme A and vitamin B5, pantothenic. Vitamin B5 is a pantothenic acid. And the two amino acid, when two amino acids are joined by a peptide bond through dehydration process, form a dipeptide. Dehydration means the removal of water molecule. The enzyme that catalyzes the formation of phosphodiester, formation of peptide bond between two amino acids is peptidyl transferase enzyme. It is also called as a ribozyme enzyme. Ribozyme is located in a larger subunit of ribosome. You know about the two types of ribosomes. One is the 70S ribosome and next one is the 80S ribosome. So in both of these two 70S and 80S ribosome, 50S is the subunit of 70S, 60S is the subunit of 80S ribosome. So this peptidyl transferase or ribozyme enzyme located in 50S of 70S ribosome, 60S of 80S ribosome, because they are the larger subunits. They are useful for the synthesis of protein by making of peptide bond between two adjacent amino acids. So now we will discuss about how peptide bond formed between two amino acids. For suppose if we see this is a one amino acid and it is another amino acid. In between both of these two amino acid, here carboxyl group is a donor of hydroxyl and amino group is a donor of H. OH plus H is equals to H2O. So one water molecule releases as a byproduct in this process during the formation of a peptide bond between two adjacent amino acids. When a peptide bond is formed between two adjacent amino acids, now it is called as a dipeptide. Every dipeptide has two amino acids and one peptide bond. Same way, all other amino acids are also joined by dehydration process, like a tripeptide, tetrapeptide, hexapeptide, heptapeptide, octapeptide, etc. For suppose if we see the structure of a tripeptide, here in the structure of tripeptide, number of amino acids are 3 and the number of peptide bonds are 2. If a polypeptide consists of 10 glycine type of amino acids, then the molecular formula of polypeptide is C20, H32, O11, N10. Why? Because approximately 9 number of water molecules are removed. 9 number of water molecules are removed during the formation of a polypeptide with 10 moles of glycine. So its molecular formula becomes C20, H32, O11, En2. You know about that the physical and chemical properties of all amino acids are decided by these three groups only, that are carboxyl group, amino group, and a free radical group. R group, free radical group means R group. So carboxyl group, amino group, and free radical groups are decides the physical and chemical properties of all amino acids. Based on the number of amino groups and carboxyl groups, the amino acids are divided into several types. They are acidic amino acids like aspartic acid and glutamic acid. Basic amino acids like lysine and arginine. That means in acidic amino acid, COH groups are more. In basic amino acid, amino groups are more. In neutral amino acids, both acidic carboxyl group and basic amino groups are equal in number. If any one amino acid has aromatic complexes in its structure, they are called aromatic amino acids. If the amino acids are having alcoholic group in their structure, they are called alcoholic amino acids. Few amino acids are having sulfur as a constituent in their structure. They are called sulfur containing amino acids. For suppose if we see this acidic amino acids, example aspartic acid and glutamic acid. Basic amino acids are lysine and arginine. Neutral amino acid glycine, alanine, and valine, leucine. They are the 4 different types. And aromatic amino acids are tyrosine, phenylalanine, and tryptophan. Alcoholic amino acids are serine and threonine. Sulfur-containing amino acids are cysteine, cysteine, and methionine. You know about methionine. Methionine is a very important amino acid in the synthesis of protein molecule. Heterocyclic compounds are if the heterocyclic compounds are present in the structure of amino acid, they are called heterocyclic amino acids. Example of heterocyclic amino acids are histidine and proline. Amides are carry more nitrogen when compared with the amino acids. Only 2 are the amides, that are one is a aspargine and the second one is a glutamine. And one important property of amino acids is the ZWITTER ionic form. Means a particular property of amino acid is ionizable nature of amino group and carboxyl groups. The structure of amino acid changes in solutions at different pH values because of its ionizable nature of amino group and carboxyl group. For suppose if we place an amino acid in ZWITTER ion, if we place an amino acid in neutral solution, if we place any one amino acid in a neutral solution, then what happened? Automatically the carboxyl group donate carboxyl. For suppose if we draw the diagram of general structure of amino acid may appears like this. This is a carboxyl group, and it is a amino group, and this is a hydrogen, and here free radical group. Here in the general structure of amino acid, this carboxyl group is a donor of hydrogen. It donates the hydrogen to amino group. When it donates the hydrogen to amino group, now amino group become ammonium ion, NH3 plus. By donating H to NH2, COO become COO minus. When both of these two different ions are arranged on same carbon, now it is named as a ZWITTER ionic form. So we get the ZWITTER ionic forms when amino acids are placed in neutral solution. When amino acids are placed in neutral solution, it is a very important property of amino acids, ZWITTER ionic form. ZWITTER ionic form. Thank you. Thank you. Thanks to one and all.